I'm Dr. Janina Jeff, Senior Bioinformatics Scientist at Illumina and host of In Those Genes podcast. I'm joined today by Dr. Francis Collins to discuss the role that the National Institutes of Health, or the NIH, has played in shaping the current precision healthcare landscape and provide insights into where we're headed. Dr. Francis Collins served as a director of the National Human Genome Research Institute during the completion of the Human Genome Sequencing Project, a transformative undertaking that led him to become the director of the NIH in 2009. With his leadership, Francis helped establish genomics research programs such as the All of Us program here in the United States, and the international projects such as the H3 Africa initiative. So, Francis, thank you for joining us.
Glad to be with you, Janina.
So, this year we saw scientific, clinical, healthcare communities come together at a speed we've never seen before to all combat COVID-19. And you've had a front row experience at, I would say, the biggest transformative collaboration our world has ever seen, scientific collaboration. Can you share a little bit of your experience?
Well, it has been an amazing period here, and I don't think I've ever seen scientists work harder or rally to a need more rapidly and more in a visionary way than what has happened in 2020 and the first part of 21. I remember January 10th when that sequence of SARS-CoV-2 was first posted on the internet. And Tony Fauci and I and the folks in the Vaccine Research Center made a decision. We got to go after this right now because it looks like it might be a very serious potential pandemic. And that is a step that many people thought we just sort of made it up on the spot, but you will know, and people listening to this probably know, that the background for being able to develop a messenger RNA vaccine goes back more than 20 years, and it had been bit by bit developed, becoming more practical, more reasonable. Maybe it was going to be a vaccine for MERS, and then SARS-CoV-2 came along. And the way in which that happened, just in terms of the design of the vaccine in about 36 hours, then the partnership with the company Moderna to get the first production, and basically in 63 days to begin the phase 1 trial. That is just breathtaking in terms of the speed with which that was done. And that played out then over the course of several months with other vaccines also coming along. And to get those first phase 3 trials started in July, and in December to have the data from phase 3 trials, each of them with 30,000 or more people showing incredible efficacy, 94, 95%, and a very good safety record, was almost more than we could have hoped for. But that was a testimony to the incredible dedication of people who just dropped everything and said, this is the most important thing I've probably ever done. I'm not going to sleep. I'm going to make this happen. And they did.
One of the things that I'm extremely passionate about is making sure diverse communities have science communicated in a culturally relevant way. And the NIH has also attacked the pandemic at a community level with the Community Engagement Alliance. Can we talk about that for a little bit?
Absolutely. I'm glad to talk about that because vaccines aren't going to help anybody if they're sitting on the shelf. So yeah, we have a big task now to try to reach out to communities to get them interested in taking part in the clinical trials and then interested in taking part in the vaccines. And that's what SEAL, the Community Engagement Alliance, has aimed to do. I asked 2 of my institute directors, Gary Gibbons of the Heart, Lung, and Blood Institute, and Eliseo Perez-Stable of the National Institute of Minority Health and Health Disparities to lead this. And they pulled together an incredible team across multiple states, tapping into community outreach efforts that we had been supporting for other kinds of projects and basically saying, we got to do everything we can right now about COVID-19. That has been a big contributor to the fact that when you look at those vaccine trials and you say, who took part? If it hadn't been for a big effort to reach out to diverse communities, I don't think we would have had a very good representation of the United States. But we got there, and it took a lot of work and a lot of reaching out and a lot of efforts to build trust. And that's the trust we need right now as the vaccines are rolling out, and we want to be sure everybody feels comfortable in the evidence that they're safe and effective.
And so due to the efforts of, SEAL and due to the efforts of private and public sectors coming together, we really have, done really substantial things around COVID-19. And so I wanted to ask you, how can we use this as an example to continue this in science as the next pandemic happens, as we start to eradicate other types of diseases that affect world populations?
Well, it's certainly not too soon to start thinking about the lessons learned. In fact, some of us have been at that now for a few months. This is not the last global pandemic that we're going to see. We expect there will be others. They may also be like this one, where a particular pathogen escapes from an animal host and gets into the human species and then goes crazy. And we need to be better prepared the next time than we were this time. We need not to slip back into complacency. I think we have learned this platform for making vaccines using messenger RNA is really powerful. And there's no reason we shouldn't start thinking about how to design right now and maybe even carry through preclinical studies vaccines against what we think might be the next most risky kind of pathogens, and they would probably be viruses, because that's usually what it is. So other coronaviruses, filoviruses, influenza, as we get closer to figuring out how to develop a universal influenza vaccine. We also need to think about that in terms of treatments too. And what we didn't really have over the past year is a really effective antiviral that works against SARS-CoV-2. So we need to work hard on developing those small molecule treatments as well and having them ready for the major classes of viruses. All of those things need to happen. But it won't work unless we also think about the structure of how we run these research studies. One of the things I had the privilege of doing this past year and a half is to lead a partnership called ACTIV, ACTIV, which stands for Accelerating COVID-19 Therapeutic Interventions and Vaccines. 20 pharmaceutical companies joined up with NIH, CDC, FDA, the Veterans Administration, the Department of Defense, and we worked together to try to prioritize which therapeutic agents ought to get tested. How do we combine our clinical trial networks so that they're ready to go at the drop of a hat? How do we write master protocols so that when you want to run a clinical trial, you don't spend the next 2 months sort of working out the bugs of what the endpoints are going to be? All of that really got done, and we must not forget that lesson. We need to be nimble and ready to go if those kinds of things become necessary. And your question is right. It's not just about pandemics. We have learned new ways to do research. We shouldn't forget those. Let me give you another example. This is about diagnostics. We needed, obviously, as soon as we knew this was a really bad infection, to have ways to quickly detect who's infected. Got off to a slow start, but even after it was going, most of the tests had to be sent off to a central laboratory. You had to wait to get the result back. What we really needed more of were these point-of-care tests that could be done on the spot and could be accurate and could also be reasonably affordable. So we at NIH were asked to develop that kind of a technology development program. We turned ourselves into a venture capital organization, and we basically said to those small businesses and academic labs, hey, if you've got a cool idea about how to detect SARS-CoV-2 in a nasal swab or a saliva sample, bring your idea to us. And we're going to throw you into a Shark Tank, and we're going to see what business experts and engineers and technologists and manufacturing people and supply chain people are going to say about your idea. And if it looks like, hey, this one might work, we're going to give you a lot of resources to make that happen. There are today almost 30 of these new technologies, including some that now can be used at home to do rapid detection of SARS-CoV-2. I wish we'd had those 6 or 9 months ago, but we got them now. And those technologies also are going to be amenable to lots of other applications, almost any infectious disease, but also other things as well. So we learned that. This was the kind of project that I don't think NIH had ever quite done, at least not like this. And I want to do that again with a lot of other applications for things where technology is coming along, but we could go a lot faster if we take advantage of this kind of a support system.
So when we think about the promise of the genome, when we think about how it really accelerates the way we do medicine, and more specifically precision medicine, Can you share a little bit of other initiatives happening at the NIH, perhaps maybe the sickle cell initiative or something around Alzheimer's?
So we do have an opportunity because genomics has become so powerful and so affordable to tackle diseases from a new perspective. Obviously, a big area that we're investing in is this All of Us long-term cohort study to follow 1 million Americans, get their complete genome sequences and everything else we can learn about them, and find out how does that play out in combination with environmental exposures, diet, exercise, other health behaviors. How do people stay healthy, or what happens that causes them to fall ill, and how can we best treat them? That's the sort of big granddaddy of all of the precision medicine projects. But then, certainly, as we're trying to identify new ways to develop insights that could lead to therapy for diseases like Alzheimer's, an obvious way to go and look is to just look at other genetic risk factors that might be lurking there. If we had enough sequence data, we could find them. I mean, we know about ApoE, and we've known about that for a long time, but there's a lot of other things that we can learn about Alzheimer's. So we have the Alzheimer's Disease Sequencing Project, which I was somewhat responsible for back in 2013, when it was first getting going, and which has now produced a whole list of potentially targetable genetic variants that predispose people to a higher risk. And then sickle cell disease you brought up. Here's one where I think we're bringing DNA sequencing not just to figure out risk factors, we're figuring out how to approach this therapeutically. And the whole approach that sequencing made possible, that pointed to a critical pathway in development of blood cells, is one of the most exciting things we have going right now with sickle cell. That is, there's this transcription factor called BCL11A, which turns off the fetal hemoglobin and turns on the adult. If your adult hemoglobin happens to be sickle hemoglobin, that's going to make you ill. But if you could keep that switch from happening, fetal hemoglobin will help you a lot. Keep that on. Well, that's where many of the approaches that are currently being used to use gene editing are basically aiming at BCL11A. That's just a fantastic story, and it's all built on genome technology. And now people who were really sick are walking around essentially looking like they might be cured. And that is a big word to say, the word that starts with a C, and it's called cure. But I think we can start to say that for sickle cell disease.
Yeah. And, you're a physician. I'm a bioinformatician. So finding that one base pair, is really difficult, right? And one of the things I learned very early on in my academic career, we were doing GWAS. And at the time, some of the first GWAS had a few hundred people. I'm like, wow, and we're doing the entire genome. Wow, this is big data, And now here we are in 2021, and GWAS, we're in the next generation of GWAS, looking at polygenic risk scores and trying to understand how those single bases come together cumulatively to help us understand disease risk. But as you know, it takes a very large sample size, and that sample size has to be extremely diverse. So when the All of Us program was announced, I was so excited because at the time, I think the biggest data set, most diverse data set I had worked with was maybe 20,000 samples. Can you tell us the promise that All of Us is holding? One of the most exciting things about it is its aim at diversity. And one of the things I'm extremely excited about is its engagement with community. Can you tell us about the promise, some of the challenges and the things that you're learning along the way?
Yeah, All of Us got slowed down by COVID-19 as far as the recruitment, like everything else did. But there are about 370,000 people already signed up on the way to 1 million. And it is the most diverse cohort ever assembled. It has 51% people of racial and ethnic minorities. And if you add to that the kind of diversity of other sorts like socioeconomic status, living in rural communities, places that are often neglected by researchers, you get up to about 85% of the participants in this group are not the traditional ones that you would see in other studies like Framingham. So we have this incredible group of people, and they're all consented in a fashion that they really understand that they're part of a national adventure here, and that they're not just along for the ride. They're our partners. And certainly they are not what you used to call human subjects, a term that I really don't like. So we and our 370,000 partners are trying to figure out, how do genes and environment and health behaviors and individual decisions, how do they play out in health? And they are agreeing to make available their electronic health records. They fill out lots of questionnaires. They give blood samples. And this is a database that we want to be available to all qualified researchers so that we can all learn from it. That is unprecedented, to have that rich kind of a data set that we can actually involve lots of smart people trying to sort out what is it that keeps people healthy. How do you take a polygenic risk score and fit that into all kinds of other things like lifestyle, environmental exposures, and really figure out what we should be advising people about how to stay healthy? I see that as such a big part of the future. Because we've gotten to the point now where you could sequence a genome, you all listening to this know this really well, for maybe $500 or $600, which means it's not that ridiculous to imagine that all of us would have our genome sequences placed in our medical record, but we still aren't quite sure what to do with it. In many instances we know, but in a lot of instances we don't. How does pharmacogenomics really work in the real world? All of us is going to help us with that too. That's the path we're on, to try to take the promise of genomic medicine and make it applicable to everybody, not just circumstances where you have a puzzling condition, you think maybe it's a genomic condition, or it's a cancer case and you want to see what the nature is of that cancer mutation in that person. Those are incredibly important. But to be able to apply it to a healthy person and say, here's the opportunity you have to stay healthy. It's not the same as the person next door. This is for you because it's based on your genome as well as all this other information, all this metadata about you that is going to assist us in giving you the best possible advice about how to stay healthy. That's a dream I've had for more than 20 years, and this is the path to make it come true.
Yes. And, one of the things the All of Us program attacks, it really helps us understand this issue of sample size and also kind of like diversity. But we know to really, really dismantle health disparities, it's an entire ecosystem. And that ecosystem includes addressing structural racism. And it includes increasing the amount of diverse voices we have who are actually doing the research beyond participants, like you said, becoming collaborators, becoming scientists. What are some of the things that the NIH is doing to really increase diversity in the workforce? We've seen a couple of papers, such as the Cell paper that was published earlier this year, that talked about the inequity of Black scientists compared to their white counterparts. What is the NIH doing to combat some of these issues?
Janina, I'm glad you're bringing it up because this is a critical issue. 2020 will be remembered, of course, for COVID-19, but I think it will also be remembered as an opportunity where many who had kind of convinced themselves that we were past racism in the U.S. realized that we were not, and that the 400 years of racism that have followed slavery are still having its aftermath around us all the time. All you got to do is look, and there it is. The killing of George Floyd opened a lot of eyes to the reality of that. We at NIH, looking at that, became immediately convinced that we were part of the problem and that our own workforce, our own community has also been in some ways affected by these 400 years of structural racism. And it's up to us to try to see how we might address that. So we have, in fact, over the course of the last 9 months, assembled the boldest group of thinkers that we could at all parts of the workforce. Some of these are senior people, some of them are graduate students and postdocs, and asked them, what should we be doing that we're not doing? In several places. One is our own workforce is not anywhere near as diverse as it should be. We should look like the United States of America. We don't. We're woefully underrepresented in African Americans and Latinos and Native Americans. The other is, of course, what is our health disparities research agenda right now? Is it really tackling the fundamentals that are undergirding all of these disparities, which fall into this category of structural racism? Or are we sort of tiptoeing around the edges? Are we continuing to actually catalog reasons for health disparities? Or are we trying to mount interventions that might work? We are determined to change our direction. And yes, we do have inequities. We are deeply troubled that an African American applicant for an NIH grant award has a lower likelihood of getting that award, even when you balance for every other parameter. And we are determined to fix that. We have limitations because of certain Supreme Court justice decisions, but we are aiming to figure out how best to get this balance back to where it should be. And we're going to have to come up with new ways to do recruiting. How do we make the biomedical research community seem like a welcoming place for all the talent that's out there in diverse communities, but which doesn't necessarily see all that much of a welcoming view or people who look like them? We're going to work on that in new and bold ways. And we have this program called Unite, UNITE, which has, I would say, probably the most aggressive approach that NIH has ever mounted to try to tackle this, including a lot more resources to go into workforce recruitment and into health disparities research.
I'm so excited about that. And I can tell you, being a Black scientist, I definitely understand the part you mentioned about wanting to be engaged and wanting to be in these environments. And it's really important that we create an environment that's safe, that welcomes everyone's culture, that embraces everyone's culture, because it truly is also an opportunity for us to learn about each other, appreciate our differences, and then scientifically, we really push change. We're asking questions that are culturally relevant. What are some of the things that the NIH is doing? And what are some of the things that we can look forward to in regards to human genetics and the promise it can hold going forward in the future?
I want to talk about the global opportunities. We are one family. Our humanhood is shared by all those other billions of people in other countries. And if there is a bit of evidence that's extremely compelling about that, it's the genome. You can look at the genome and say, oh my gosh, we are all so closely related. And yet our studies in genomics and the access to healthcare are not evenly distributed. I want us really in this next decade to take genomic opportunities further in addressing problems in global health. And one of the areas that has taken, I think, a lot of momentum and run with it is H3 Africa, the human health and heredity in Africa that was started, and I had a bit to do with that about 12 years ago, and which is now, I think, spinning off a whole new area of data science in Africa, which can build on some of that genomic information. But right now, what we know about the genome is still awfully overrepresented for people of European background. And we need to fix that. And now that this has become so accessible and affordable, that's something we should do. We're going to learn a lot that way about biology and about medicine, and we should not be limiting ourselves. And we also, as people who are part of the global family, should feel some responsibility to take some of the advances that are happening in genomics and export them to low and middle income countries that could benefit from them as well. Right now, for instance, why is it that it is so hard to get a complete viral sequence from somebody who's just got infected with SARS-CoV-2 in Uganda? That ought to be something we could do like that. I'm calling on all of my global health buddies, and I know a lot of them are probably listening to this. Let's really do what we can here to speed up the exportation of genomic capabilities to places that need that. So that's one of my goals for the next 10 years. I also want to see, as I told this little example about progeria, the more we can move our capabilities from diagnostics into therapeutics and even into cures, we should do everything possible to make that happen. And that means investing in approaches that you can kind of see might work, but really pulling all the smart people together to do so. How are we going to take in vivo gene editing to the next step? There are 7,000 genetic diseases where we know the precise DNA misspelling. If you had a way to zip code your gene editor and get it right to the right tissue and appropriately programmed to fix that mutation, you'd have a scalable approach to those 7,000 diseases. You wouldn't have to tackle each one at a time, which might take us another century. We should push on that. Talk about a triumph for the genomic approach. Right now, the triumph is that we can diagnose, but it's going to be a lot more exciting if we can take those diseases and offer them something like a treatment or even a cure. So another dream that I've got. And we're starting there with places like sickle cell disease, but we should extrapolate that as fast as we can.
So is there anything inspirational that you could tell our audience that they could think about that maybe could help us continue on and push forward with this large collaboration across industry, across academia, globally? What is something inspiring you want to leave with everyone?
I think all of us got into this field of medical research and technology development because we want to help people. We want the human condition to be improved by our work. We want people to have the maximum opportunity to live out not just a good lifespan, but a good health span, where illness doesn't cut down people prematurely or limit their possibilities. So imagine that that's our goal. How do we get there? And it's not all about health, of course. All the economic needs, the educational needs, those have to be folded into maximizing human flourishing. But you could say health is just about the center of this issue. Well, okay, what's in the center of health? It's basically coming up with strategies to prevent disease and to treat it when it happens. That's medical research. So medical research is kind of in the center of the center. Well, what's the central technology, the central approach now that's going to get us there? I think I'm going to argue it's genomics. That basically that is so fundamental. That's reading the instruction book for that person in order to benefit them in a way that otherwise you might not have been able to do. So yeah, if you imagine my concentric circles now, genomics ends up being the center of the center of the center. And if you believe that's so, that is both incredibly exciting and it's also a heavy responsibility. Because if that's the promise, if that's the place where more people across this planet are going to have a chance to flourish, we got no time to waste. And that means bringing together all of the smart ideas, all of the partnerships, all of the ways in which we could go to that next level of applications and waste no time. Let's just get it done.
I couldn't agree more. So thank you so much. This has been great, amazing. Definitely a bucket list item for me. It was great to meet you.
Nice to meet you too, Janina.